Hello everyone. My name is Laura Tilley. I work for the CTAF General Secretariat, and I was kindly invited by the CTAF to give Paleocene fossil wood from Antarctica, based previously because my supervisor Jane Francis gave a talk on paleontology in the polar regions at the CTAF 50 General Assembly. So my PhD was completed in 2016 at the University of Leeds, and it was funded by the British Antarctic Survey and the National Environmental Research Council in the UK. And here are my supervisors, Jane Francis, who is currently the head of the British Antarctic Survey. So I'm just waiting for the screen to change. It might be a bit slow. Oh, nope, too fast. Okay, so before I go into the details of my PhD research, I'd like to provide some background on the Paleocene climates and geography. So the Paleocene epoch is considered an interesting analogue for understanding the possible effects of future climate change because it was a greenhouse world. Well, let me start off by saying, actually, just for you who don't know, who are not have a geological background, that the Paleocene epoch stretches from 66 to 56 million years ago, and this was a period directly after the extinction of the dinosaurs. And yes, it's an interesting time for understanding the possible effects of future climate change because it was a greenhouse world with little or no ice in the polar regions. The latitudinal temperature gradient was more shallow. Latitudinal gradient was more shallow than today. The CO2 levels were similar or higher than today. And you can see that there was evidence of crocodiles and coal deposits within these high latitudinal regions. The paleogeography was similar to present day. Antarctica was situated over the South Pole, but instead of being covered in ice, the Antarctic Peninsula was covered in dense, cool, temperate forest, similar to the forests found in Chile, New Zealand, and South Australia today. The continent was still joined by land bridges to South America and to Australia. And these forests are now preserved as fossil wood, leaves, and pollen. So moving on to the aims of my PhD and the objectives. So the aim of my PhD was to reconstruct the Paleocene forests and climates of Antarctica using fossil wood. The objectives were to identify tree types that lived in these forests and to use tree rings and cell anatomy of the wood to reconstruct climate features such as annual temperature, rainfall, and seasonality. And this was to understand how the climate systems worked at high latitudes within this greenhouse world. So my supervisors collected the fossil wood that I studied from Seymour Island, which is situated on the northeastern tip of the Antarctic Peninsula. Seymour Island is totally barren. There's no ice, which means that there's full rock exposure, and these fossil flora can easily be found. So you can see at the bottom of the screen, I don't know, I'm just checking that you can all see the screen because I noticed some boxes flying around. This is a picture of some fossil wood and it's exceptionally preserved. This is a picture of some fossil pollen which is also found in the sediments of Seymour Island. And to give some background on the geolith, so to the west, the Proto-Pacific Pacific Ocean plate was subducted underneath, and to the east was a marine Podocarp basin, and the sediments on Seymour Island represent the evolution of this Podocarp basin. So the trees that were growing on this volcanic arc, once they died, the wood was transported via river systems into this marine basin, where it floated around as driftwood, and then eventually it became saturated and sank to the bottom of the sea, where it became permineralized and preserved. And often within the fossil wood, you can see the borings of marine organisms, But just to reiterate that this slide shows the stratigraphy of Seymour Island, the Paleocene sedimentary succession.
Slides aren't sharing at the moment as well.
Okay. Oh, wait, it's not showing. Okay. Let me just get out of this. Get to the right one. Okay. Yeah. So this diagram is the sedimentary succession or stratigraphy, sedimentary log from which the wood was collected, and it youngs upwards. So the younger rocks are at the top, and it represents a shallowing sequence. So a shallowing sequence of that basin from a marine shelf environment represented by fine sediments and silts to a more fluvial terrestrial environment represented by more coarse sediments and fluvial sedimentary structures associated with rivers or debris flows. And the wood is all allochthonous, so it's not situated in its original position, as I mentioned before. There are different types of preservation. So there's calcite permineralization, more associated with the marine preservation. Some of the wood is rounded and charcoalified, which suggests that this may have been transported from further inland in upland areas, where it was prone to where it's prone to areas that were prone to fires from volcanic activity or possibly lightning strikes. Another type of preservation is iron oxide, and this is associated with more terrestrial depositional environments. So possibly the wood represented from this formation, the Cross Valley Formation, may represent forests from more inland areas that's traveled a shorter distance. So the wood was collected and the exact location on the sedimentary log was recorded, so that the taxonomic information and the climate information could be put into the exact stratigraphic context, so the exact time context, as much as possible. So before moving on to my results and the types of fossil wood that I identified, I will give you an overview of wood anatomy, so I can orientate you in the diagnostic features that I use to identify fossil wood. I'm just going to move this screen out of the way. So within my fossil wood sample, there is conifer wood and there is angiosperm wood, so flowering plants. And conifer wood is made of tracheids. They're these long tube-like cells that carry water throughout the tree. And at the start of a growing season, these tracheids are quite wide and large. And then towards the end of the growing season, they become more narrow with thicker walled cells. And this change in diameter creates the growth ring, which you can see in this image to the left, the transversal view. On the walls of the tracheids, you get, there are bordered pits, and the arrangement of these bordered pits and the shape and the size are very diagnostic for identifying fossil wood. And the purpose is to control water flow from one tracheid to the next. So in this radial view, you can see ray cells which cut across the tracheids, and these ray cells store and carry nutrients throughout the tree. And where those ray cells intersect with the tracheid cells, these are called cross fields, and within these cross fields are pits or circular, or well, they can be any shape. And the number of pits within these cells and the shape and the arrangement is also a very diagnostic feature for identifying conifer wood. And if you look at the tangential section, so you can see the end of the rays and the length of the rays in terms of cells, and the width, so the number of cells in width, can also be a diagnostic feature. So this is conifer wood, but moving on to angiosperm wood, which is slightly different. So instead of tracheids, angiosperm wood has vessels, and these vessels carry water throughout the tree. And they are supported by fibers. So the fibers give the mechanical support to the vessels. And if there are different arrangements of these vessels, which are diagnostic for different types of wood. So there is a type of wood called diffuse porous, and this is where the vessels remain the same size throughout a growth ring. Or there is ring porous, and this is where the vessels become smaller towards the end of the growing season. And then within the walls of these vessels, you get intervessel pitting, and the arrangement and the size of these pits are very useful in identifying wood. And within the vessels, you also get these perforation plates, and they can be a simple hole-like structure or a ladder-like structure, and they control the water from one vessel to the next. And again, similar to the conifer wood, in the radial section, you get these ray cells running across the vessels and the fibers, and where the vessels and the ray cells intersect, there is something called vessel ray pitting, and this vessel ray pitting is very useful in identifying wood. And similar to the conifer wood in tangential view, you can see the end of these rays, and the length and the width can also be useful. So that's just a very brief introduction to fossil wood anatomy. To identify the fossil wood, or any wood, the wood has to be first cut into those three sections: the transversal section, the radial section, and the tangential section. And then thin sections are made of these sections, so thin that you can see through them and light can be transmitted through them. And then they are viewed through a petrographic microscope, and then systematic measurements are made on these various features to identify the wood. So just moving on to the next slide. Takes its time. So moving on to the types of wood that I identified. So the first type of wood is a conifer wood, and its morphogenous name is Agatha oxlon. And its characteristic features is that it has very distinct growth rings. The bordered pits on the tracheids are very close together, and they can be 2 to 1 width apart, or even 3. And if you look closer, there's a more zoom picture, they have an alternative pattern. So they're very close together, and they have an alternative pattern to them. And also the crossfield pits are very unique to Araucaria wood, very obvious, very close together, and have an alternative type of arrangement to them. Or alternate, rather, would be the best word for that. So its nearest living relative is Araucaria, or Agathis, and today they live in warm to cool temperate forests in New Zealand and Chile, and yeah, well, mostly New Zealand and Chile. And they live at, well, they grow at mostly high altitude, in high altitude areas. They are they're shade intolerant, so they live sorry, let me take some water. Yes, they're shade intolerant, so they live on the at the tree line, in very exposed areas with little coverage of other plants, and they rely on disturbances such as volcanic activity to rejuvenate and to clear the surrounding vegetation so they can regrow. And they're often found with southern beech, Nothofagus wood trees. So the second type of wood that was identified was a conifer wood as well, part of the Podocarp family called Phyllocladoxylon. That's the morphogenous name. And the features of this type of wood is that they have bordered pits, usually single bordered pits on the tracheids, and they're usually separate, not joined together. And they usually have one large crossfield pit within the rays, as you can see here. The modern relative is Phyllocladus, which is mainly found in New Zealand, and they have a wide tolerance of altitude, so from low altitude to high altitude. They're shade tolerant, so they can grow in closed forests, but they're also found in more alpine areas, and they prefer poorly fertile or poorly drained soils. So the first angiosperm wood that I identified was Nothofagus, and the diagnostic features of this wood is that they have vessels that are arranged. So this is the top view looking down at the growth ring. So they have vessels that are arranged in a more radial type of pattern. They have perforation plates that are simple, so circular, and also ladder type perforation plates. The intervessel pitting, so I just need to say that this wood is exceptionally preserved and looks, I'm just reminding myself that it looks like modern wood. The anatomy is still very well preserved, considering it's 56 million years old. So yeah, it has these opposite intervessel pitting arrangements, so aligned, if you like, and it can be a bit elongated and oval shaped, and also the intervessel crosswheel pits are oval shaped, but its modern relative is the southern beech tree, and these are found in cool temperate forests in New Zealand, Chile, and Tasmania in Australia. And they prefer mid to high altitude areas and forest borders and more exposed woodland. They are a shade intolerant tree. So like Araucaria, they prefer to live in areas of high disturbance, such as areas of where there's volcanic activity, where it clears the surrounding forest and they can regenerate. It's known as a pioneer species, so it's usually the first to establish itself after such a disturbance. So another angiosperm tree is Myja Suginella oxon, and yes, it has distinct characteristics in terms of solitary vessels rather than joined together. It's diffuse porous, so the vessels don't really change throughout the growth ring. It has mostly scalariform perforation plates and elongated vessel ray pitting. Its modern relative is Luma, or Myja, and this is found in Chile, in the Valdivia forests of Chile. And it's usually found in low to mid altitudes, and is shade tolerant. It's a canopy or subcanopy tree. And finally, the last angiosperm wood that I identified was Windmania oxon. And yes, this is characterized by mostly solitary vessels and scalariform perforation plates, and this ladder-like vessel ray pitting, so very elongated vessel ray pitting and joined rays that you can see in tangential view. So the end of the rays are usually joined to each other. And its modern relative is Windmania. And this is again a low altitude to mid altitude tree, and it's shade tolerant to shade intolerant. So yeah, this is the main. I think it likes to grow in well-drained soils. So yeah. So now moving on to the methods that I used for reconstructing the Paleocene climates of Antarctica using the fossil wood. So the first method is growth ring analysis. So all the fossil wood that I studied has distinct growth rings, which suggests that these trees grew in a seasonal climate. And I measured the growth rings, the width of the growth rings of each of the specimens. And from the series of the growth rings measured, for each specimen, I calculated the mean, and that gave me a mean growth ring width for each specimen. So basically, a short growing season equals narrow growth rings, and a long growing season equals wide growth rings. And another method used was mean sensitivity, and this calculates the variation in growth ring width from one season to the next, so from one growth ring to the next. And it's an indicator of the variation in growth conditions. So whether the climate was stable or it was always variable from one season to the next. Another analysis used was on angiosperm wood, and it's vessel analysis. The methods are called the vulnerability index and the mesomorphy index, and they are indicators of a plant's adaptation to the availability of water within its environments. And they are based on the observation that trees that grow in arid environments tend to have vessels with a smaller diameter and thicker walls, and vessels that are more frequent and shorter vessels. So to calculate these two indices, I measured the vessel diameters, the vessel abundance, and the vessel length. Another method used was specific gravity, and this was calculated mathematically, and it's based on the thickness of the cell walls of the wood, and it provides an indication of the ecology and the growth conditions of the wood. And yeah, so it's indicating of plant growth, the mechanical strength of the wood, and the hydraulic capacity. The final method used was something called the coexistent approach. And this is an approach to reconstruct past climate conditions on the assumption that fossil flora had the same climate tolerances as their modern relatives, which we'll go into a bit later. So I'm just changing the presentation again. So just before, so I also did a bit of pollen analysis to complement the research on the wood, just to get a fuller picture of the floral content in these Antarctic forests, and to see if there was different plants observed, because the pollen record also represents non-woody plants, and it's very well preserved. Again, it looks exactly like this pollen was deposited yesterday, and there are some new types of plants observed. So yeah, we have the Nothofagus also represented. Also this podocarp called Huon pine, and it's native to Tasmania, and it grows along rivers. Also different types of podocarps. There's Microcrachis, and this is a podocarp shrub. It's also native to Tasmania and grows in alpine areas. And yeah, there was lots of fern spores, so tree ferns and other small angiosperms like Proteaceae, which is found in quite a lot of places that has a wide range, climate range. So I also did this to increase the data set for the coexistence analysis, looking at the climate tolerances. So yeah, moving on to the coexistent approach. So basically this graph, it just shows an example of how it's done. So you take the climate tolerances, so in this case mean annual temperature ranges for the nearest living relatives of your fossil assemblage, and then where they all coexist provides the range in temperature. So the results from doing this type of analysis was that I got a mean annual temperature of 7.7 to 13 degrees, and a mean annual precipitation of 1,000 to 2,489 millimeters per year. So this suggests that the climates on the Antarctic Peninsula during the Paleocene were cool to warm temperate, and there was sufficient water all year round. And the temperature and the precipitation is similar to the climates of New Zealand, Chile, and Tasmania. So moving on to the mean growth ring width analysis. So I'm sorry that this diagram is a little small, but it's directly from my publication. And it took me a long time to plot. So basically it shows the stratigraphic log. so going from oldest to youngest, and the symbols represent the different wood types, and the numbers within the symbols are just the specimen number. But this analysis, first of all, it looks a bit noisy, the data, and the reason for this is that, well, they are different wood types. Well, let me say the purpose of doing this analysis was to see how climate changed from the early Paleocene to the late Paleocene, but the data is quite noisy. It's not a linear pattern, and the reason for this is that the different tree types that have different growth habits and possibly come from different environments on the Antarctic Peninsula, so they have their own environmental constraints. But we can see that there appears to be a faint pattern for showing that from the beginning of the Paleocene, moving to the early late Paleocene, there is a decrease in growth ring width, which suggests possibly a change to cooler and drier climates. And then at the very top, it starts to, the growth ring width gets a bit wider, so possibly a change back to warmer, wet climates at the end of the late Paleocene. And this graph is just to show, I took an average of all the wood from the different sections, so across this section, and it appears to show the same pattern. But I am aware that it could also be an artifact of the sampling, because towards the top of this sedimentary succession, there's only a few wood types, a few wood data points, and they are of a few wood types, so that might be biased towards that type of tree type and its growth habits as well. So I'm just changing it. I'm waiting for the screens to change. So this is the results for the mean sensitivity analysis on the growth rings. And again, we have the stratigraphic log with the data points plotted against it to get to see the evolution of climate through time. And so mean sensitivity, so values below 0.3 suggest that the tree had a complacent growth, so growth under an equable climate where there was no variability in resources from one year to the next. And then sensitive growth, which is values above 0.3, and this shows that there was some variability in growth conditions. And the near majority, I think it's almost equal, show equable climate conditions, but there is a fair bit of wood that shows sensitive growth. And it's interesting that these wood types that show sensitive growth are mostly Agathocxlon and Nothofagus. And this may suggest that they had similar life habits to their nearest living relatives. So they lived in they may have lived in upland areas near the forest borders at the very limits of the threshold, which were very exposed areas, which means that the precipitation or the availability of resources may not have been effective, would have been less affected due to being more exposed and being affected by temperature and wind and solar radiation. Also, sensitive growth, it could be due to areas prone to disturbance, so that can cause variability. So this could suggest that they had a similar similar to their nearest living relatives, but there is generally no trend to this data through time, so it's a bit all over the place. So again, so this is the vessel analysis results. So I forgot to label it. So on the far left side, we have the vulnerability index method, and on the right side we have the Mesomorphy index method, and we'll call it VI, mostly shows growth under arid conditions, whereas the MI shows that there was sufficient water availability for the wood or for the trees growing on the Antarctic Peninsula. And there is some contradictory information, even with specimens. So specimen number 3 that I've circled in green, the MI shows that there was sufficient water availability. And then in the VA analysis, it shows arid conditions. But there has been recent work done that shows that the method, the Mesomorphy index, it has a closer link to water availability than the vulnerability index, because it uses more vessel variables or characteristics. So it includes vessel length, where the VI does not. And then, so the last method, specific gravity. So the same situation. So we have low specific gravity, 0.4 and below, then medium, and then high specific gravity. And there's no trend through time, but most of the woods fall within medium specific gravity. And this is expected for a temperate forest. Usually from reading studies, tropical trees, you have a wider variety of specific gravity. And then the wood showed the black triangle, which is Phyllocladus wood. This is a very high specific gravity, and its modern relative also has a very high specific gravity, even though it lives in an environment with sufficient water availability. And the reason for this is that it's very slow growing, so it has a because it's slow growing, it has a more thicker celled wood rather than a tree that is fast growing, which would have a lower specific gravity because the wood is less dense. So this analysis just provides a bit more information on the ecology, and the wood possibly had similar life habits to the modern relatives. So moving on to the summary of my work. So this is a summarized diagram of my data plotted along with previously published climate data. So along the side, we have the stratigraphic formation, so the section and the time. Across the top, we have temperature, and along the bottom, we have the mean growth ring width. And first of all, my data, which is shown for the mean growth ring width data, which is shown by a black dot. This is mostly agreeable with previously published work by Francis and Poole that also looked at mean growth ring width through the Paleocene. And this is shown by the white dots, but it's quite hard to directly compare them because the wood from their study, we're unsure exactly which level it was taken from, but it generally, they both show from the start of the Paleocene a cooling trend. So a narrowing of growth ring width, which shows possible cooler or drier conditions. And then in the Cross Valley Formation, so towards the Late Paleocene, this changes to warmer environments. So in terms of temperature data, my temperature ranges are comparable or in general agreement with temperature data derived from proxies from geochemical analysis on organic matter that creates temperature data, and also mean annual temperature data from other fossil floral analysis from Antarctica. So yeah, so my work is in general agreements, and in the wider context, the cooling trend that is observed in my growth ring data is consistent with global climate data derived from benthic oxygen isotope records, which is an indirect indicator of temperature. So there was a move towards heavier oxygen isotopes through the early Paleocene to the late Paleocene, which is suggested the formation of ice on land. So just to give you sort of a visual overview of how the forests of Antarctica may have looked. So this is a painting by artist James McKay from the University of Leeds. And this is a reconstruction painting of actually the forests on Antarctica during the Cretaceous, so before the dinosaurs went extinct. So just forget about the dinosaurs, but the flora was similar. So we have the Araucaria living in the hinterland, high altitude areas, and then we have like Huon pine in the Proteaceae, and some Nothofagus and Podocarps living in lowland areas along the river. So it's quite a nice reconstruction to try and visualize what it may have looked like on Antarctica during the Paleocene. So finally, the last slide. So my study represents a new detailed and robust reconstruction of terrestrial Paleocene climates of the Antarctic Peninsula using newly studied fossil wood assemblages from Seymour Island. The tree types identified are characteristic of the trees living in warm to cool temperate forests in New Zealand and Chile, Chile today, and Tasmania also. The fossil wood represents trees possibly originating from different altitudes on the Antarctic Peninsula. And it's possible that the Araucaria and the Nothofagus trees, especially the ones that showed sensitive mean sensitivity, may have had a similar habit to the nearest living relatives and possibly grew in highland areas. The growth ring analysis shows a trend towards cooler climates from the early Paleocene to the early late Paleocene, and then warming again towards the end of the Paleocene. And generally, the vessel analysis shows that there was sufficient water availability, and so does the coexistent approach. And the specific gravity values also suggest a temperate forest environments, or they were growing under temperate climates. Okay, yeah, so this is the end of my talk, and I hope that you enjoyed it. And I'm open for any questions.
Do we raise hands or can?
Yeah. Oh, yes. Okay. Yeah, Patricia.
Yeah, sorry, I wasn't there at the start because I had a other meeting. I was just wondering, as you are working on wood, are you collaborating with the Xylarium in Tervuren, which has these 60,000 samples and thin slides and so on from all over the world?
No, but
Okay.
no, but I'd like to do something with this.
Okay.
But I'm due to publish. Yeah, I'm due to publish this work soon. So once I've published, I will look for some little project on the side to do there possibly.
Okay, so I would really recommend, even if I don't know if you will continue to work on this, but it's really worth a visit to see the collection. So you can contact Hans Beekman from Tervuren.
Yep, great, thank you. Thank you. So Carolyn?
Yes, Laura. Thank you very much for your presentation. So I really know what you are doing, even if it's a long time that we are meeting each other in the institute. I have a question concerning the plants that are not trees, that are not forest. Are there information, for example, from the pollen of what are the other plants that were living in Antarctica in these periods?
Yes, there's actually my supervisor, who's a palynologist. She published a paper in 2015 identifying the different pollen that was on Antarctica from the same succession as my wood. And yes, they've also show similar information, like similar climates. So pollen is not as conservative as fossil wood. Fossil wood, it's hard to know the species, or to say that this is a definite species. But pollen, you can have a better idea. For example, fossil wood that is a podocarp, I can just say that it's a podocarp. But the pollen shows that there was different types of podocarps, like Huan pine or Microcrassus. So it gives a more detailed, also a detailed look into these different plant types. Yeah. Did that answer your question?
Yes, but so there are other species and also than trees that they discover with the pollen.
Yeah, yes. Also, lots of different ferns as well, and and mosses. There's actually quite a few mosses that were found in the pollen sample or the spore sample, pollen and spore sample.
Thank you.
Okay, thank you. This is from Ole Kansandra, I think that's how I pronounce. How many growth rings did you measure, and what was the diversity of the fossil wood, presumably? So altogether there was 56 specimens, and I tried to take wood that had a long series of growth rings, so it's more reliable, so it gave a more regional signal. So that's usually the best is a specimen with 50 growth rings or more. And the composition of my sample, there was, in terms of angiosperms, there was over 50%, I would say, was Nothofagus, Southern Beech. There was a lot of Araucaria. So Araucaria, Nothofagus, also Phyllocledoxon wood was also, these were the most prominent wood types in my sample. Yeah. Okay. Thank you for joining this to listen to my presentation. And yeah, I will publish my work soon. And if there are no more questions, I will let you get on with your day, and thank you very, very much.